Hello everyone, welcome to biology class. In this class we shall see 12th standard Botany, chapter 4, Principles and Processes of Biotechnology. The next topic of this chapter we are going to see green fluorescent protein. In the previous part of the video we have seen some of the products of biotechnology. So in this class we shall see about green fluorescent protein. So this GFP is a protein which contains 238 amino acid residues and its molecular weight is 26.9 kilodalton and it exhibits bright fluorescence when exposed to blue to ultraviolet range, that is 395 nanometer. And this GFP, green fluorescent protein, it refers to protein isolated from the jellyfish. Okay, so this protein is isolated from the jellyfish Aquaria victoria, and this is an excellent tool in biology because it has an ability to form internal chromophore. Okay, that is the color production. They have a protein through which it can exhibit a bright green fluorescence, and this does not require any accessory cofactors, gene product, enzymes or any substrates other than molecular oxygen. So in cell and molecular biology, this protein gene is used as reporter of expression, and it can also be modified to make biosensors. Now let's see a protocol how to insert the gene into another plant. Okay, so this green fluorescent protein which is isolated from the jellyfish, after gene splicing technique, we can alter the codon and this can be introduced. This protein can be introduced from this plant. The protein can be taken and it can be introduced into Arabidopsis thaliana plant. And now this plant, okay, Arabidopsis thaliana plant is known to exhibit the fluorescence. The next topic is biofarming. Biofarming is otherwise known as molecular farming, and it is used for the production and use of transgenic plants genetically engineered for production of pharmaceutical substances for the use of human beings. And this is otherwise referred to as molecular farming or farming. And these plants, they are different from the naturally available medicinal plants. And we are using the plant systems as bioreactors. And many pharmaceutical substances can be produced using transgenic plants. Example, golden rice, which we have seen in the previous video. The next topic is bioremediation. So bioremediation is nothing but a method in which microorganisms or plants are used to clean up environmental pollution. And using this approach, we can treat the waste, including wastewater, industrial waste and solid waste. And this process is applied to the removal of oil, petrochemical residues, pesticides or heavy metals from soil or groundwater. This method is less expensive and it is more sustainable than other methods of remediation. And this method is cheaper and eco-friendly method also. And this method can be used to deal with the lower concentration of contaminants more effectively. The strategies used for bioremediation are, so in this process they are using indigenous, that is the natural microbial population as indicator for bioremediation process. So this bioremediation with the addition of adapted or designed microbial inoculants. So it can be done even with the natural microorganism or after designing the microorganism also this can be done. And we can also use, apart from microorganism, plants can also be used for bioremediation, which is referred to as green technology. Some examples of bioremediation technology. Phytoremediation, phyto meaning plant. When we use plants for remediation, it is called phytoremediation. So plants used for cleaning up the environmental pollutant. Mycoremediation, myco refers to fungi. So when fungi is used for treating the environmental pollutant, it is called mycoremediation. Next, bioventing. Bioventing is a process that increases oxygen or airflow to accelerate the degradation of environmental pollutant. So by using this bioventing, we can increase the oxygen level, and thereby we can degrade the environmental pollutant. Leaching, bio-leaching. So using microorganism to recover metal pollutant from contaminated sites. Next, bio-augmentation. Augmentation is nothing but speeding up process. So we can use the selected microbes to speed up the degradation process. Next is composting. So composting is nothing but the process by which solid waste can be composted by using the microorganism, and the waste can be composted in the form of a manure, and this manure can be used as a nutrient for plant growth. Rhizofiltration. It is the uptake of metals or degradation of organic compound by the soil microorganisms. And rhizostimulation is the stimulation of plant growth, okay, by reducing the toxic materials. Next, limitations of this bioremediation process. So this bioremediation process can be employed only to the biodegradable contaminants. And this process must be specifically made in accordance to the condition. So whatever the contaminated site condition prevail, this process must be made in accordance with that conditions. Small scale test on a pilot scale must be performed. Okay, so before performing a large scale production for treatment remediation process, small scale test has to be performed. And use of genetic engineering technology to create genetically modified microorganism for bioremediation process. Next, biofuel, or otherwise called algal biofuel or algal fuel. So this is an alternative to liquid fossil fuel, that is the petroleum product. And this use algae as a source of energy. And algal fuels are an alternative to commonly known biofuels which are obtained from corn and sugarcane. The energy crisis and the world food crisis have initiated interest in algal culture. Okay, so that is for farming algae for making biodiesel and biofuel using the land unsuitable for agriculture. And Botryococcus brownie is normally used to produce algal biofuel. So how it is produced? The biological hydrogen production by algae. The biological hydrogen production with algae is a method of photobiological water splitting. So in normal photosynthetic process, the water is splitted, okay, and thereby during the photosynthetic process, the plants will release oxygen. Okay, but here, instead of the process of releasing oxygen, if, for example, Chlamydomonas reinhardti, if it releases oxygen under normal condition, when it is deprived means made unavailable of sulfur, it switches to the production of hydrogen. Instead of oxygen, it will start producing hydrogen. So this hydrogen can be used as a fuel. Okay. So this is called as hydrogen production by algae. And here the electrons are transported. Okay, in the normal electron transport chain, the electrons are transported to ferredoxin, and the ferredoxin hydrogenase enzymes combine them to the production of hydrogen gas. The next topic is bioprospecting. So it is the process of discovery and commercialization of new products which are obtained from biological resources. So it may also involve biopiracy, so in which the indigenous knowledge, that is the natural knowledge originating with the indigenous people, is used by others for profit without authorization or compensation. So when they are using the profit or indigenous substances without the knowledge of the indigenous people, then it is called biopiracy. So it is manipulation of intellectual property right laws by corporation to gain exclusive control over national genetic resources without giving adequate remuneration or recognition to the original possessors. An example is of biopiracy. The recent patent granted by US Patent and Trademark Office to American companies on turmeric, neem and basmati rice. So these are all indigenous crops of Indo-Pak subcontinent, but they have got the patent right. Okay, so this is an example for biopiracy. First topic, biopiracy of neem. So the neem, we the people of India are using this neem and its oil in many ways for controlling the bacterial and fungal skin infection. And we are more generous to share the information, that is the properties of the neem, with the entire world. But this knowledge has been pirated, and United States Department of Agriculture and an American MNC, they have sought a patent from the European Patent Office on the method of controlling of diseases on plants by the aid of extracted hydrophobic neem oil. And the patenting of the fungicidal and antibacterial properties of neem is an example of biopiracy, but later the traditional knowledge of the Indian was protected. The next is biopiracy of turmeric. The United States Patent and Trademark Office in the year 1995, they granted a patent to the method of using turmeric as an antiseptic agent. And this turmeric has been used by Indians as home remedy for quick healing of wound and also for the purpose of healing rash. And the journal article published by the Indian Medical Association in the year 1953, this remedy, that is in the year 1953 itself, the remedy was mentioned, that is using the turmeric for healing of wounds, that is as an antiseptic agent. But the United States Patent Trademark Office in the year 1995, they have granted a patent for them for using the turmeric as an antiseptic agent. Therefore, in this way, we have proved that, okay, so the use of turmeric as an antiseptic is not new to the world, and this is not a new invention, and it has been a part and parcel of our own traditional knowledge of India. And so the objection in this case, US Patent and Trademark Office was upheld, and at last the traditional knowledge of the Indians was protected. And this is also an example for biopiracy. The next is biopiracy of basmati. So on September 2, 1997, the US Patent and Trademark Office granted patent on basmati rice, lines and grains to the Texas-based company Rice Tech. Okay, this patent gives the company several rights. That is the exclusive use of the term basmati as well as the proprietary rights on the seeds and grains from any crosses. And the patent also covers the process of breeding Rice Tech's novel rice lines and the method of determining the cooking properties and starch content. And India has perilled the United Nations to take the matter to WTO and as an infringement to the TRIPS treatment, which could have resulted in major embarrassment for the US. And hence, voluntarily and due to few decisions taken by the US Patent Office, Rice Tech had no choice but to lose most of the claims and most importantly the right to call the rice basmati. And the year 2002, the final decision was taken, and the tech dropped down 15 claims, resulting in clearing the path of Indian basmati rice exports to the foreign countries. And the patent office ordered the patent name has to be changed as Rice Line 867. The last topic of this chapter, applications of biotechnology. So biotechnology is the most important applied interdisciplinary science of 21st century. And it is a trusted area that enables us to find the beneficial ways of life. And this has a wide application in different sectors, agriculture, medicine, environment, etc. And this science, the biotechnological science, has an invaluable outcome like production of transgenic varieties of plants, cotton, rice, tomato, tobacco, cauliflower, potato, banana, etc. And the development of transgenic, apart from transgenic varieties of the crops, it also imparts some additional values to the plant crops such as pesticide resistance, stress resistance, disease resistance, etc. And even for the synthesis of human insulin and blood protein in E. coli and can be utilized for insulin deficiency disorder. It is also a big breakthrough in the field of medicine. And the synthesis of various vaccines, enzymes, antibiotics, dairy products, beverages, they are also the product of this biotech industries. And biochip based biological computer, okay, so which is also very important in the field of biotechnology. And genetic engineering, Okay, so which involves genetic manipulation, tissue culture technique, how to develop the totipotent plant cell. So everything is also an added on application of biotechnology. And even the single cell protein. Okay, so it is obtained from spirulina, and few other microorganisms is also employed for production of SCP. So it is also a application of biotechnology. Apart from that, many secondary metabolites can be produced, various biofertilizers, biopesticides, enzymes can be produced. Okay, and other techniques such as biomass energy, biofuels, bioremediation, phytoremediation. So all these aspects play an important role in environmental biotechnology, and these are the innumerable applications of biotechnology. So with this we come to the end of this chapter. Thank you. Meet you in the next video.